Welcome back, folks. This is video lecture chapter 16 on genomics, the whole genome perspective. The chapter presents a fantastic perspective on how current genomes have come into existence. It compares them between organisms to see if there's a pattern, and it attempts to extract the secrets of how genomes are related to their RNA and protein functionality. The chapter is organized into 4 parts, the first of which tackles the relationship between the genome and its genes. The first obvious takeaway is that there is an enormous variation in the size of a genome across living organisms. Some organisms, such as bacteria, only have a single DNA chromosome. Others have hundreds of chromosomes, such as plants, fish, and amphibians. The number of genes generally is correlated to the complexity of the organism, but the genomes also vary in their proportions of coding versus non-coding sequences, and this ratio reveals some fascinating departures from what we've just said. The most efficient way to determine the arrangement of genes and other functionality of a chromosome is to sequence the DNA. Traditional DNA sequencing methods were very cumbersome and expensive, but new, latest technology, such as nanopore sequencing, is making it so much easier that within a single day we can easily sequence an entire genome multiple times. Traditionally, primer walking was a reliable method of successfully gathering information on the sequence of a DNA molecule, and this was based on generating a sequence, then near the far end of that sequence making a primer that will be unique, and then doing a further sequencing experiment and then walking forward, repeating this procedure again and again until you got to the end of a chromosome. An alternative strategy used in the past also was that of shotgun sequencing, and that relied on breaking the DNA into fragments of workable size and then sequencing those fragments again and again, and hopefully there would be some overlap between the fragments that would allow the computer to match one fragment with another. These techniques relied on a library of sequences being generated in the first instance, and then the computer would take over and form contiguous sequences along the entire chromosome, and this was known as contig mapping. Table 16.1 gives us a fantastic insight into the relationship between different organisms. So the simplest organisms are placed at the top and some of the more complicated ones at the bottom. And then each column gives us some data on the size of the genome, the number of genes, and the level of density of genes along the DNA. So looking at this data here, we can see that simpler organisms have a lot of genes per megabase of DNA, whereas more complex organisms tend to have fewer genes per megabase of DNA. In the case of primates, we're down to about 6 to 7 genes per million bases. Most of the current data that we have on genomes of organisms relied on 2 general methods, which are listed here. The first was whole genome shotgun sequencing, where the DNA of the entire genome is fragmented in some way, and those pieces are chosen at random and sequenced. Or the alternative, clone-by-clone sequencing, where the chromosomes once again are broken into overlapping clones and are then arranged in linear order to produce a physical map. Thereafter, the clones are sequenced separately to determine the sequence of DNA. The second method has a caveat in that it relies on existing resources to enable the researchers to apply this technique. So only some model organisms are amenable. Unlike the whole genome shotgun approach, WSG, where any organism is subject to this technique, no prior construction of physical maps or anything else is necessary. The entire genome is broken into fragments, sequenced, and the resulting sequences are assembled into these contigs. Hopefully we have overlap, and it's the overlap component that's the most important. If there's no overlap, then the computer is unable to connect one fragment with others. The gold standard amongst geneticists is to sequence the same region of DNA 30 to 40 times in order to make sure that we have full coverage. Figure 16.1a shows us how primer walking works. Suppose you want to sequence this region in blue. You design a primer to some part of that sequence, maybe the brown on the outside, and then you sequence inwards. Once you have a confidence in a part of that sequence, you design another primer, and then you walk further inwards from both sides to save time. And eventually you'll come to a point where there's an overlap between your walks, and that should be the sequence of that region. In B, we see the example of shotgun cloning. Shotgun cloning generates lots of fragments through a random process that we'll talk about shortly, and then these fragments are sequenced only for their ends, the regions in red. The computer overlaps them to produce a contig. Now sometimes there may be gaps in the contig, but those gaps can be closed by further sequencing, or other techniques such as PCR walking. The real challenge to WSG comes with repetitive DNA. This is especially true of large repeats on the chromosomes, which are bigger than your ability to generate these contigs. Ch16.2 illustrates this conundrum. Suppose the colored regions, the blue, the brown, and the purple, are unique along the chromosome. The yellow regions are repeats, and they may be repeats like each other. So when you generate the contigs and you try to put them together, the possibilities are based on how the computer tries to line up the regions in between. So that will confuse the computer, and it will generate a multitude of options, only of which one would represent the real situation. A solution to this challenge has been developed, and that's called paired end sequence data. In paired end sequencing, the fragments are first generated in a very broad library, a library containing large fragments. Those fragments are cloned into cloning vectors, the ends are sequenced, and then they are fed into a computer. The goal here is that the repeat sequence would be part of the clone. I think this is illustrated nicely in figure 16.3. So these 20 to 30,000 base pair fragments are hopefully large enough to encompass the repeats in yellow. So by doing their ends, you now know the ends of this fragment, and because the fragment came from a chromosome, there is no shuffling of DNA. So this end is physically connected to that end. Then, in order to get the information for the middle parts of these purple and blue fragments, you generate those data from a library made of smaller fragments that are then sequenced. The computer will then take this information and make these contigs. But when it comes to this data in the middle, those fragments cannot be orientated because of the challenge. But we compensate by using the information from these clones, clone X to fill this gap, and clone Y to fill this gap. You could use the bottom clone too, because it will confirm that these ends are connected to each other. So this data can be utilized to confirm the orientation of this contig. There was one caveat with paired end sequencing, and that was if the size of the fragments was smaller than the repeat, then this method could not be used. But as we keep saying throughout this chapter, the latest technologies, the latest DNA sequencing technologies, they have no issue with repetitive sequences as far as we know whatsoever. From a historical perspective, the very first entire bacterial genome to be sequenced was of the bacteria Haemophilus influenza. And it has a 1.8 million base pair circular DNA with few dispersed repetitive elements, so it was conducive to sequencing. The sequence data was obtained by fragmenting the bacterial chromosome into 3 genomic libraries that consisted of 140 contigs, and the computer was able to put them together, and very few gaps were left at the time, which have now since been completed. Using the technology of the day, 140 gaps were unfulfilled. Of those, 98 were sequence gaps, and 42 two were physical gaps. Newer methods were able to overcome the 98 sequence gaps, so those were completed soon after. The 42 physical gaps had no clones to sequence. These physical gaps were closed using two methods available at the time. One was to use large lambda genomic libraries for probing the sequences from the ends of the scaffolds, and the other one was to use PCR, and those techniques became available soon after. And that led to the complete sequencing of the genome of this bacteria. The entire challenge of sequencing Haemophilus influenza is presented in Figure 16.4a. Figure 16.4b gives us an annotated view of the chromosome showing where different genes of different types are located along the entire chromosome. Since we're talking about repeats making it difficult to sequence DNA, the very first eukaryotic genome with significant number of repeats to be sequenced was from the fruit fly Drosophila. Its genome is 170 million base pairs long, with 120 million base pairs of euchromatin and 50 million base pairs of heterochromatin. The heterochromatin did present problems for the researchers, so they focused their initial efforts on the euchromatin, and then only later, when techniques became available, they were able to tackle the heterochromatin. Other than that, the techniques used were whole genome shotgun sequencing. The holy grail of sequencing is, of course, the human genome. This project was spearheaded by the Department of Energy under the guidance of the National Institutes of Health and other outsourced projects around the world. This began in 1990, and the clone-by-clone approach was determined to be the best method to generate the physical map of the human genome initially. During the project in 1998, Craig Bentner and his company, Cellular Corporation, began to use whole genome shotgun sequencing approach. The projects were completed in 2001. The two draft sequences were published on the same day, and they contained many gaps, but those gaps have subsequently been filled by using newer techniques. Incidentally, some of the DNA included in this project came from Craig Ventnor himself. However, he wasn't the only human being to contribute to this project. That leads us to reference genomes. Reference genomes are genomes of a single individual, either haploid or diploid, that are sequenced, and their DNA profile is the gold standard for that species. Any differences, such as polymorphisms between individuals, are then aligned with the reference sequence to be determined. So the evolutionary history of a species is reflected within those polymorphic alleles within the population being studied. These projects have revealed the littering of our genomes with various types of polymorphisms ranging from single nucleotide polymorphisms, SNPs, to inversions and deletions and translocations, and even in some cases with some organisms, whole genome duplications. With respect to the human genome, the number of SNPs varies, and we believe that 30 to 50 new SNPs are introduced into each germ cell per generation. With respect to an alternative type of polymorphism, that of copy number variants, these can range from small areas of DNA to huge tracts of DNA, some of which are hundreds of thousands of kilobases in length. And any genes that they encompass will also be present in multiple copies, and that can give rise to the phenomenon of gene dosage, where the products of genes are out of balance with respect to the rest of the genome. Insertions and deletions, on the other hand, are the result of unequal crossing over during meiosis, and they can lead to repetitive sequences appearing because of misalignment. Thus, the lowest common denominator when it comes to individuals of a population would be called the pan-genome. Every member of that population would be expected to have this as its base. Then on top of that, you could have additional copy number variants or repetitive sequences or other types of polymorphism. Figure 16.6 puts into perspective this relationship between these copy number variants. And as we said, they can range from a single nucleotide change, SNPs, all the way through to entire chromosome sets being copied or missing, including chromosomes. And we learnt this in a previous chapter. The lower half of the figure simply shows you how misalignment can lead to both increase and decrease in the number of these repeats. Suppose you went down to your local sewer and took a sample of the liquid from that location, and you sequenced everything that you found in terms of DNA within that solution. That would be a metagenome. So a metagenome is a collection of DNAs from that environment that represents all the creatures that occupy that environment, and it also tells us the proportion of that environment that's occupied by different organisms. So metagenomics is a study of the environmental community that exists at any location, and this could be inside your body or outside your body, in a pool of water or in the atmosphere. Generating the sequence of DNA is relatively straightforward these days. Once you have that data, the next task is to assign functions to the different segments of the chromosomes, and that is called gene annotation. So gene annotation is the process of attaching biological function to DNA sequences, and this is based on experimental data and also computational analysis. And our computers are becoming more and more powerful at helping us. If one recalls, only about 2% or less of our genome is responsible for coding for protein. The remainder is involved in generating regulatory sequences. Many other parts are just repetitive DNA based on transposons and retroviruses. And there's a small segment of the DNA whose function we have still yet to decipher. All these activities will be under the title of DNA annotation. At the next deeper level, the identity of each gene needs to be tallied to align these genes to either a biochemical function, a cellular function, or a biological function. A number of successful strategies have been employed in the past, such as using cDNA to compare to genomic sequences, which allows one to identify transcribed regions of the genome. In a perfect world, if one generates a cDNA library that represents truly the complete set of messenger RNAs present inside that cell, then one should be able to annotate all the genes of that organism over time. But in reality, that's not the case, because no single cell has all its genes represented as messenger RNA at any one time. Figure 16.7 allows us to see the activity of one type of annotation, that acquired from experimental data, that shows how genes can be annotated. So by using data from partial or full-length cDNAs, we can come up with this annotation here, where we see the untranscribed region, the start codon, the location of intron-exon boundaries, and the 3' untranslated region, as well as the stop codon. These approaches are very successful when computers are brought into the picture. Once you combine biology with computational power, you come to the field of bioinformatics. In bioinformatics, the computers are programmed to look for patterns that indicate open reading frames. An open reading frame is simply a long region of DNA that codes for proteins that suggest that it may be part of a gene or a gene. The computer software can be programmed to search either using strict criteria or loose criteria. One starts with loose criteria, where you tell it to look for a minimum size of amino acids. Because of experimental data, most proteins have 50 plus amino acids as part of their polypeptide sequence. For any segment of DNA, the gene could lie on either strand. Thus, the computer software takes a starting position, first going one way and then on the opposite strand going the other way, by looking at each base and counting that towards a codon, and then shifting to the next base and doing likewise. So for the bottom strand, 3 different reading frames are examined, and the same thing for the top strand. So a total of 6 reading frames are interrogated by the computer. Now, these programs have to take into account what's really out there, and what's really out there are the presence of introns of various lengths and interrupted genes that can cause some ambiguous data to be generated. At this time, there's no substitute for using experimental data rather than using computer predictions. In the future, as computers become more powerful and AI, artificial intelligence, begins to be more reliable, the outcomes may change. Open reading frames are always looking for protein coding genes. But we now know that the genome contains far more DNA dedicated to non-protein coding genes. These include the structural genes for tRNA and ribosomal RNA, and spliceosomes, etc., and the regulatory RNAs like microRNAs and pbRNAs. In these cases, computers have a hard time finding matches. The primary goal of gene annotation is still to ascribe a biological and biochemical function to genes, in addition to their location and their structural organization. As an example, the LAC-I gene from the bacteria and its involvement with the lacopron has led to a lot of experimental data being generated, and that experimental data allows us to annotate that gene in a lot of detail. If we discover other genes with similar layouts or similar sequences, we can conclude that they may have similar biochemical outcomes for a cell in which they reside. Functional genomics is the field of biology where we want to assign or ascribe a function to each region of the DNA if possible. And in this, combined with annotation, has been a very powerful source, but a science that's still being worked out. Regardless, the results are pretty impressive. In figure 16.8a, we're looking at Arabidopsis, the weed, and we see that its genome has already, for the most part, been classified, even though about 45% of it remains to be classified. Contrast that to figure 16.8a, the genome annotation of Drosophila, the fruit fly, with predicted biological function, and we can see clearly that over 50% of the genome has unknown, unclassified function as of this time. One component of gene structural analysis that helps us is that genes fall into gene families, and these gene families have arisen by evolutionary mechanisms. Therefore, they still retain some resemblance to each other in terms of function or structure. With respect to the human genome, we know that there are 21,000 protein coding genes, and those roughly 21,000 genes can be classified into 10,000 gene families, so about 2 genes on average per gene family. Some gene families may have 4 more members than others. Others may only be present in single copy. A great example is the olfactory receptors, whose gene family in humans numbers 400 members or so. But in other creatures, such as mice, these numbers could be up as high as 12 to 1400 members. This segues nicely into protein domains. Students sometimes have a hard time with secondary structure of proteins, alpha helices and beta sheets, tertiary structure of proteins, localized folding, and this thing called protein domains. Protein domains are simply functional or structural regions of a protein that are independent of other regions of that protein. Protein domain hunting is made easier by comparing proteins with similar types of domains that can then be ascribed to a particular function. Many of these protein domains seem to be preserved over genomes. So if you find a new region of chromosome and it resembles a protein domain, the chances are that it may have a function related to the original. Figure 16.9 associates modularity with protein domains. In panel A, EPC-like protein is used as an example. So evidence suggests that the ancient protein consisted of 2 EP domains and 2 PhD domains. Since then, the PhD domains have been kept together because they may have a particular module that they form that's very important in binding to, say, DNA. The position of the EP domains has also been preserved, although you can see that they have been modified with additional domains that have been brought in by recombination. The bottom figure compares different categories of organisms, eukaryotes, with protein architectures. The general takeaway from this is that multicellular eukaryotes have more complex protein architectures than single-celled eukaryotes. The more data that we have acquired, the better the analysis that we can perform. Because we have so much data from bacteria, archaea, and eukaryotes, one avenue of investigation is to compare all three to draw some general conclusions about genome organization. One such conclusion is that on average, bacteria and archaea have fewer genes per genome, and their gene density is much higher than in eukaryotes, mainly because they lack introns, but also because they have very compact gene regulatory sequences. They don't have the enhancers or the silencers in distant places as we do, and their overall lower complexity of protein structure in terms of domains. Amongst eukaryotes, their organization differs widely with respect to gene number and gene density. In general, single-celled eukaryotes tend to have fewer genes than multicellular eukaryotes. The lifestyle of the organism then further influences its genome organization and gene content. So obligate parasites that must live on their hosts often experience something known as genome contraction. They lose some of their gene functions because those functions are provided by the host. Figure 16.10 compares the genomes and the gene organization amongst E. coli, a yeast, a plant, an invertebrate, and a vertebrate such as us. One major discrepancy across eukaryotes is the amount of repetitive DNA that their genomes carry. This seems to be related to the particular branch of the tree of life along which that organism has evolved. The human genome contains over 50% of its DNA as repetitive DNA. And out of that 50%, 45% are transposable elements, 3% are microsatellites, and a further 5% represent gene duplications. That's not including the telomere and centromere sequences that exist. Repetitive DNA not localized at these telomeres or centromeres has a name, and that's called dispersed repetitive DNA. Figure 1611 shows one of the smaller human chromosomes, chromosome 21, and you can see that by using different techniques, we are able to localize and annotate that chromosome in various ways, from broad to very specific. So in conclusion, by comparing genome sequences from many organisms, that has led us to make three basic conclusions. Genomes are dynamic. They can change over time. Genome sequencing of model organisms reveals limitations of forward genetic screening. And the number of genes in the human genome is comparable to that of other multicellular eukaryotes, so there's nothing special about humans. Section 16.3 delves into evolutionary genomics. Evolutionary genomics is defined as the comparative study of genomes. Some sources call this phylogenomics or comparative genomics. One could compare organisms within the same species. That will be known as intraspecific comparisons. Or one could compare between species. That's called interspecific comparisons. Both of these yield important data. Based on DNA sequence, it's turning out to be one of the most powerful tools available to us to predict not just the present, but also the past. more important it appears than fossil data. Even though one can't replace the other, the reliance on DNA data is in most cases more valuable than fossil data. In particular, scientists have been using ribosomal RNAs as the model for investigating the origins of life and any relationships that exist between existing species. And they have come to a startling conclusion that life can be divided into three domains: bacteria, archaea, and eukarya. This figure, 1612, clearly shows the relationship based on all the evidence presented in this textbook. We can see clearly that eukarya are more closely related to each other than are organisms of archaea. But archaea are more closely related to eukarya than either is to bacteria. But so far, there are clearly three individual domains separated from each other. The logic behind such comparisons is straightforward. DNA tends to acquire mutations. Since these mutations are rare events, most of the time the DNA doesn't change. So by aligning using computers the regions of DNA that are homologous, then we can ascertain the phylogenetic relationships as a result of these mutations. So homologous nucleotides are those that are descended from the same nucleotide in the common ancestor without change. That brings two species together. Various landmarks are utilized, such as the highly conserved protein coding DNA sequences, and those landmarks are known as nodes in the branch points of the tree of life. Freely available computer software, such as BLAST, enable these alignments to be performed very, very quickly. These analyses are very, very powerful because by aligning sequences across species, we're able to deduce their evolutionary relationship to each other quite succinctly. The logic is simple. Two closely related species that have evolved more recently should retain a large proportion of their DNA sequence in homology. Figure 16.13 shows the results of one experimental match. In this, 4 different species of Saccharomyces, including our friend Saccharomyces cerevisiae, are compared for a stretch of chromosome. The blue boxes represent open reading frames. The dotted lines correspond to orthologous open reading frames. That means these must have come from an ancestor. And in blocks that are red, we can see that certain common open reading frames may have then collapsed to produce a smaller gene compared to the wild-type original ancestral sequence that exists. Studies such as this reveal that differences do exist, and there may not be any real reason or rhyme as to how the differences materialize, but large tracts of DNA still retain the homology that came in from the common ancestors. How do these gene rearrangements take place? How do new genes arise and some genes disappear, become deleted? The mechanisms that we have seen in other chapters apply here too. Gene duplication, by having extra copies of chromosomes, can lead to an amplification of both the DNA content and also the gene output. An alternative way to get gene duplication is by unequal crossing over amongst the existing DNA and chromosomes. Exon shuffling leads to new combinations of genes being produced, and a more recent powerful discovery is that reverse transcription can lead to the generation of new mechanisms of gene structure. The birth of genes is depicted for each one of those mechanisms graphically in figure 16.14. See if you can relate to this by your information knowledge from previous chapters. But wait, there are other mechanisms that also can lead to the birth and death of genes. So some genes are derivatives of exons from transposons. Others are due to horizontal gene transfer, lateral gene transfer. Gene fusion and gene fission are also responsible for breaking up and making new genes. And sometimes new genes are created de novo in place. The lower half of figure 1614 represents that in a graphical sense, and once again, see if you can relate to each one of these mechanisms. You have seen them before, but do you still remember them? According to data from Drosophila species experiments, the major source of new genes, slightly less than 80% in this case, was gene duplication. The driving force for these gene duplications was in some cases about 10% of the time due to retrotransposition, and in about 12% of the cases they arose de novo from non-coding sequences that turned into genes. These duplicated genes were found either in tandem or dispersed throughout the genome. Most genomes contain a mosaic of gene families derived from ancient or recent duplication events. So which is more common? Well, a study published in 2000 by Lynch and Connery estimated that the duplication rate to be about approximately 0.01 gene per million years, which can be restated in a different way that for an average eukaryotic genome consisting of between 10 and 30,000 genes, that would include humans, this would be one gene duplication event per 3 to 10,000 years. Once a region of DNA containing a gene becomes duplicated, what's the outcome of those two or more duplicated genes? Well, that depends on the molecular basis of the duplication. If the entire gene, including its regulatory sequence, is duplicated, both copies can continue to produce functional product in the correct amount, time, and space, and there is no major dosage effect of these two genes now working together in the genome, then the genes are redundant and they're free to evolve new functions. The evolutionary pressure on that genome will force one of three outcomes. In the majority of cases, the new gene degenerates and becomes a pseudogene, because the other copy of the gene is enough to maintain the organism. This extra copy then accumulates mutations and it renders it non-functional in the vast majority of cases. These mutations could be in the coding region or in the regulatory region. We call that a lack of positive selection. If mutations accumulate in both genes, there may be a case where one gene complements the activity of the other gene to restore the biochemical function back to that cell, and we call that subfunctionalization. In some rare cases, a mutation in one copy of the gene may provide it with a new function that it didn't have previously, and that's called neofunctionalization. Figure 615 tries to relay the fates of these duplicated genes across these three probable outcomes. Please make sure that you fully understand the meaning behind the gene and its biochemical function. This slide just reiterates some of the terminology that's very important when one starts comparing across evolutionary timelines the relationship amongst genes. Homologs or homologous genes are genes that descended from a common ancestor, and they constitute a gene family. Paralogs are genes that originated from a duplication event, performing biologically related but distinct functions for that cell. Orthologs are genes in different species that are derived from a single common ancestor. In most cases, these orthologous genes are equivalent in function due to descent from that common ancestor. The human beta-globin gene cluster is a fantastic example showing orthology and paralogy involving the transmittance of genes from our common ancestor to both humans and chimpanzees. We learned in a different chapter that lateral gene transfer is also known as horizontal gene transfer. So the transfer of genetic material between two existing species, not related to their descent. And a common mechanism is the sharing of plasmids. By all estimates, about 1.5 to 15% of genes for any genome are the result of lateral transfer. This is more common in prokaryotes than eukaryotes, but it still happens to some degree across eukaryotes. The evidence suggests that certain genes are more prone to lateral transfer than other classes of genes. What we're trying to say here is that genes for biological metabolic functions are the ones that tend to be more readily found as a result of lateral horizontal gene transfer than are genes for information processing functions. One barrier to successful lateral gene transfer are the differences in biology between prokaryotes and eukaryotes. Although the one prominent exception is the endosymbiosis of chloroplasts and mitochondria that are now part of every eukaryotic nuclei. Very few lateral gene transfers between eukaryotes are known. If one discovers an unknown area that resembles a gene, by studying the same sequence in a related species and knowing its function there, one can apply that knowledge to this original species. Mutation is a random process. Areas of the genome that have a defined role are less likely to tolerate mutations than areas of the genome that are non-essential. Therefore, by comparing a number of different organisms that are closely related, one can identify quite quickly regions that code for genes or regulatory elements away from those regions that are not, finger quotes, as important. The computer programs, as we mentioned earlier, are designed to seek all possible reading frames in all directions. So 3 on one strand and 3 on the other. There are several criteria under which gene annotation fails. One is the lack of homology to known genes. The other is the existence of small genes or exons that can be difficult to predict using computer programs, and or the breaking of genes into multiple small exons that code for fewer than 100 amino acids. Those can be tricky for computer analysis software. CNSs stands for conserved non-coding sequences. So if that gene doesn't produce a protein product, it will fall into this category. That would include regulatory sequences or genes that produce functional RNAs. There are several approaches that have been developed to address and discover these regions. Those are included in these 3 gray slides where they talk about phylogenetic footprinting and phylogenetic shadowing. You may read these at your own leisure. Let's move on. The next genetic term that we need to be familiar with is word synteny. Synteny simply tells us that there's a conserved order of genes along a piece of chromosome. So if A, B, C, D exist in a chimpanzee, then A, B, C, D will also exist in the same order in a human. That would be then synteny. Synteny is normally a label applied to large stretches of chromosome that maintain integrity between species. Another label called microsynteny is applied to low levels of chromosomal arrangement involving just a few genes that have been seen in some analysis. Figure 1619 shows synteny between human and mouse chromosome equivalents. The order of the genes is almost the same across this patch of chromosome between these 2 species. Why do humans have 2 copies of every gene? In some cases, we have 4 copies of every gene. In exceptional cases, we can have 10 copies of every gene. Well, the answer seems to be due to 2 mechanisms of genome duplication. One is small sections of chromosomes can become duplicated, and the other is whole chromosome sets can become duplicated. By comparative phylogenomics, one has come to the conclusion that in the past, whole genome duplications have taken place, as well as many smaller duplications for parts of chromosomes. When whole genome duplication events lead to duplicate sets of genes, we call these homologs, with the word E in the middle. Homologs. Plants, in particular, and amphibians, and some fish species reveal whole genome duplications. Panel A suggests how a haploid creature can become diploid in a single generation if its chromosomes do not segregate as expected during mitosis or meiosis. The second half of this figure shows the chromosomes of Arabidopsis, the weed, and how different regions of those chromosomes have become duplicated over time and maintained throughout the genome. The final part of the chapter looks at functional genomics and its goal to elucidate gene function. The rate at which computers are populating our databases with sequence data is astonishing, and it continues to grow exponentially year on year. Once that data is available, then it has to be annotated, as we said in the previous section. How do we get from one to the other? How do we assign function to those gene segments in terms of development and physiology? Well, what do we need to know? Well, we need to know when and where these genes are expressed. And we need to know the consequences in terms of mutations of these genes. Do they provide a gain of function or a loss of function? We also need to know, are these genes part of a redundant pathway? Or which proteins each gene product interacts with eventually? These types of answers are the domain of functional genomics, which is the study of gene function from a whole genome perspective. Instead of looking at each gene individually, a large collection of genes are simultaneously analyzed to see if they produce expression patterns, interactions, or other biological findings. One such approach is the field of transcriptomics, which is defined as the study of gene expression from a genomic perspective. As with cDNA libraries and their reliance on messenger RNA, in this case, the entire set of transcripts from the DNA present in a cell, or even the entire organism at that particular time, is called a transcriptome. Two commonly used methods, one of DNA microarrays and the other of high-throughput sequencing of cDNA, are used to answer these questions, with the latter becoming the dominant method because of the ease at which DNA can now be sequenced. The sequencing method involves the following steps. RNA is isolated from these cells and converted to cDNA, as we mentioned in a different chapter. The cDNA is fragmented and sequenced using high-throughput sequencing, although the latest sequencing technologies do not need the DNA to be fragmented, so saving a lot of time. Regardless, the resulting sequence, known as an RNA-seq, is compared to the reference genome of that species, which allows one to identify sequences present in the cDNA population. As with cDNA libraries, there are two advantages to this technique. The sequencing has a potential to become more quantitative. That means we can tell which cDNA fragments are more common in cells than others. This correlates back to which transcripts are more prevalent than others. Second, further in-depth knowledge of which transcript of a particular gene is more common compared to other alternative transcripts has been obtained. Using these methods, many of the non-protein coding transcripts have been identified. The alternative method using DNA microarrays consists of using chips, silicon chips, as in computers. Instead of using resistors and transistors, the scientists use DNA that's synthesized and affixed to each location on this chip. The population of these molecules could represent an entire genome, or part of a genome, or whatever the scientist wants to add to that chip. In most cases, these fragments that are synthesized are between 25 and 70 bases long. They're not double-stranded because they have to hybridize to your DNA of interest. There has to be a mechanism in order to detect which fragments have hybridized. And normally what happens is that the experimental DNA is tagged with fluorescent probes at one end of the DNA, and the intensity of that signal is measured by laser beam and detected by a computer. Figure 1621b shows a diagram of a part of a DNA chip. Each of these regions contains a unique set of DNA fragments, all identical on each spot. The computer is aware of where each of these maps to the genome. During the experiment, RNA is extracted and then converted to cDNA, and that cDNA is made as single-stranded before it's labeled with a fluorescent tag. And then the two are incubated together. And regions where the DNA attaches are identified by fluorescence. The different colors correspond to different levels of expression. Alternative chips can be made that carry expression arrays rather than genomic DNA. So these chips carry a DNA synthetically attached to each spot that represents one of the many arrays discovered for that tissue. Hybridization with labeled cDNA produces quantitative information about the relative expression levels of every gene that's been tested against that array. These chips are so powerful that they can also be used to identify DNA binding sites, as well as proteins already bound to the DNA. This is referred to as the chip sequence when one applies this technology to chromatin immunoprecipitation. The chips are so powerful that they can give a single glance at a genome-wide protein-DNA interaction relationship. One great example of success using these techniques has been published with Saccharomyces cerevisiae. These haploid cells, they normally undergo sporulation, and we thought that they used 150 genes in order to direct the sporulation process. Now that was based on forward genetic mutational analysis. More recently, genome-wide expression analysis using these chip techniques, considering the same type of biological process of sporulation, has uncovered more than 1,000 genes involved in sporulation. Indeed, more recently, it's becoming common for doctors to analyze the transcriptome of cancer cells in order to determine which genes are morphologically defective in that one cancer, thereby potentially tailoring the treatment to the patient's cancer. Figure 1622, even though the detail is not available on this slide, it does show you how the pattern of gene expression varies with the time of sporulation. Let's put all the pieces together in this animation from the textbook.
When a complete genome sequence is obtained for an organism, functional genomics research can begin to describe the functions of all genes in the genome. Measuring the levels of messenger RNAs, for example, indicates the global gene expression state of the cell. The total set of messenger RNAs present in a cell at a given time is known as the transcriptome. The transcriptome changes significantly as the cell responds to its environment and as it carries out normal cellular events, such as growth and division, or, in the case of yeast, sporulation, as shown here. Defining what genes are expressed, when they are expressed, and their levels of expression is key to obtaining an understanding of cell functions at the global level. DNA microarrays are commonly used for studying changes in gene expression throughout the life cycle of an organism. For example, Pat Brown and Ira Herskowitz used DNA microarrays to study changes in the transcriptome of yeast during sporulation, the process of producing haploid spores by meiosis. Here, we will present a simplified version of their experiments to demonstrate the utility of DNA microarrays for such studies. First, 6,200 protein-coding genes representing 97% of the genes of the yeast genome are amplified using PCR. This DNA is denatured and spotted onto the glass substrate at known locations. Diploid yeast cells growing in normal medium are harvested and used to create a control sample. To create this sample, the mRNA is isolated from the cell. This mRNA is used to synthesize cDNA molecules using reverse transcriptase. To tag this DNA, the DUTPs used in the reverse transcription mixture are labeled with Cy3, a green fluorescent molecule. A sample of these same diploid yeast cells are grown in a medium that induces sporulation, rather than in the normal growth medium. At distinct time points throughout sporulation, yeast cells are harvested, and their mRNA is isolated. The cDNAs from these cells are labeled with Cy5, a red fluorescent molecule. Gene expression at each of these stages can be quantified using DNA microarrays. A separate DNA microarray containing 97% of the genes of the yeast genome is used for each time point. The red-labeled cDNAs created from the transcriptome for that time point are allowed to hybridize with the DNA molecules on the microarray. However, at the same time, the green-labeled cDNAs from the reference sample are loaded onto the microarray. By loading both samples, scientists can visualize and quantify differences in gene expression between cells at each time point and cells of the control sample. A red spot indicates overexpression of a particular gene compared to the reference sample, while a green spot indicates underexpression of a particular gene compared to the reference sample. Yellow spots indicate roughly equal expression between the experimental sample and the reference sample. A black spot indicates that the gene at that location is not expressed in the experimental sample or the control sample. Laser detection can be used to quantify the amount of green and red in each spot, allowing the scientists to quantify gene expression at each time point. Using this technique, scientists have found at least 7 temporal classes of genes. In each temporal class, certain genes are overexpressed relative to the control, and other genes are underexpressed relative to the control. Knowing when genes are active gives scientists an important clue to begin to understand the function of each gene.
It appears that scientists have gone crazy with the use of the word omics or ohms. As illustrated here, proteomics is the study of all the proteins expressed in a cell, tissue, or individual. This would then represent the proteome of that cell, tissue, or individual. Special techniques have had to be devised in order to study the interaction of proteins with the DNA or amongst the proteins themselves. One ingenious method to identify protein interactions is the two-hybrid system. In the two-hybrid system, we rely on a natural system that has been dissected by geneticists for a number of years. The Gal4 gene transcription factor from yeast has a modular design to its protein, and it is this that's the heart of the method. The Gal4 transcription factor has 2 domains. It has the DNA binding domain, which binds to the DNA, and an activation domain that interacts with RNA polymerase at the promoter to activate transcription. Figure 1623a shows the natural relationship between the DNA at the promoter site, the binding of the transcription factor, its 2 parts, its 2 domains, and the association with RNA polymerase at the +1 transcription site. In this case, the reporter gene is the LAC-Z gene, which can use XGAL to then identify white colonies from blue colonies in the selection process. What scientists have done is that they've introduced 2 extra proteins in between the 2 domains of the Gal4 transcription factor. The bait protein can be attached to the DNA binding domain, and the prey protein can be attached to the RNA polymerase II binding domain. If the 2 proteins have a natural affinity for each other, they will bind, bringing the BD and the AD domains of the Gal4 transcription factor into approximate association to initiate transcription. If the two proteins don't naturally bind to each other, then that will not take place, that association, and the AD will no longer be bound to the BD by these intermediaries, and there will be no transcription. So whether transcription takes place or not is the deciding factor, and that tells us if there's an interaction between the two proteins that are being studied. The terminology used in this instance is translationally fused. So one of the proteins to be tested is translationally fused to the DNA binding domain of Gal4, while the other is translationally fused to the activation domain that binds to the RNA polymerase II. These chimeric genes, both of them, are transformed into yeast strains, and then the yeast is allowed to express these proteins in its cells. Then we can test if the two proteins interact by the presence of appropriate types of colonies on the plates. By utilizing knowledge of yeast biology in terms of auxotrophs and prototrophs, we are able to design an experiment that has the appropriate reporter genes expressed under appropriate conditions. In one such experiment, a histidine auxotroph strain of yeast is utilized. This strain of yeast cannot grow unless it can make histidine. So histidine is not supplied in the medium, but it has to be made by the yeast. Once the yeast is transferred over to the histidine plate, then it will start to die unless it can make its own histidine. In this scenario here, it can only make its own histidine if it activates the gene, this transgene, which consists of the upstream autonomous sequence for the Gal4 blended to the histidine gene. This association can only take place if the two halves of the Gal4 system carrying the bait and prey for the two proteins that are being tested will complement each other. If so, then histidine is made by the yeast cell and is able to survive and grow naturally on that plate. If, however, the two proteins, the bait and the prey, do not associate with each other, then there's no transcription of the histidine gene, and in this case, there's no growth. By repeating these type of experiments with different combinations of proteins, one is able to generate a map of how different networks of proteins interact. And the total sum of all such interactions is known as an ohm, an interactome. In this example, 10 yeast proteins were tested in various reciprocal two-hybrid experiment pairs, and we came up with this working model of how that pathway of protein interaction actually exists inside a yeast cell. The genomic method of investigation has been applied to reverse genetics. Indeed, because we start with sequence, the chromosome itself, we're able to identify much more that resides on that chromosome compared to forward genetic analysis and their screens that rely on mutations, because not every region of the chromosome may be subject to mutational analysis with this type of forward genetic screening. Evidently, to fully understand gene function, we need to analyze both loss of function and gain of function mutations. And unfortunately, forward genetics is unable to supply all the necessary components. A great starting point for such experimental analysis is a collection of mutant alleles for every gene in a genome that is known as a knockout library. We spoke about mouse knockout libraries in another chapter. In the quest to classify genes, one question is which genes are important or essential to life and which ones are optional. These questions have been answered with Saccharomyces cerevisiae. From those analyses, it appears about 20% of the yeast genes define the essential gene set. That means 186 of the deletion mutants had reduced growth phenotypes as heterozygotes and thus were haploinsufficient. Of the remaining 5,000 genes, 891 had slow growth defects that left 4,000 non-essential genes. Here is that data in graphical form. So out of the 6,500 genes present in this yeast, 20% are necessary for growth and life. The others are not essential. 80% are not essential, 20% are. What does it mean to be non-essential or a non-essential gene? Simply means that under certain growth conditions, those genes are not expressed or not needed. By changing the environment, different genes out of that 4,000 would become important. One major outcome of this experiment was that 200 genes could not be assigned any function. Did this mean that they're truly non-essential, or that there were testing conditions outside the scope for which these genes may be needed? Or did they have overlapping functions as other genes? Or simply that the scientists that annotated these as being genes were incorrect? We'll have to wait and see. Additional research of the type just mentioned has come up with more types of interactions that exist inside real living systems. Gene interactions themselves can give us clues as to how a particular gene functions in vivo. Studies with double mutants, where two genes are affected by mutation and studied together, has revealed that there are a multitude of genetic networks that operate. One finding from these studies is that of synthetic lethality. In these cases, if one of the two genes is mutated, then that's not lethal to the cell or organism, nor is the other gene mutated by itself. But if both genes are mutated together, that results in death, and that is what synthetic lethality is. The outcomes of these experiments are astonishing. They suggest that within these genetic networks, a small number of essential genes are participating in a multitude of reactions. And in addition, that more non-essential genes are participating in fewer interactions. That data was put together and it gave rise to this type of graphic. Looks like something the CIA would develop on terrorist networks, but the same technology is used here. So the genes labeled with a yellow background are the essential genes that are involved in many interactions. You can see 1, 2, 3, 4, 5, 6, 7, 8. And the others are the non-essential genes that participate in a 1 or 2 reaction profile. The color coding of these balls is given here. So you can see for mitosis, these genes are very important, as are these genes and a couple over here. The grouping together of different colors and their association with a core essential gene suggests that these gene networks are very real and that more has to be investigated about them. When one studies genetic interactions in genetic networks, then one is basically studying systems biology, whose primary goal is to predict biological functions. The ultimate goal for functional genomics is to find the role of every single gene in every single circumstance in an organism. And we're far from that goal at this time for many creatures. Thank you very much.